Welcome to Hima Learning Academy. Today, we are breaking down the science of RT-PCR and how we detect RNA viruses. There is often a bit of confusion in the lab. While RT can sometimes mean real-time, in this process, it specifically stands for reverse transcription. This is the essential first step, where we convert viral RNA into a format that a PCR machine can actually read. Step 1 is RNA extraction. In the lab, we use specialized lysis chemicals to break open the viral envelope and release its genetic material. As you can see, the single-stranded RNA is isolated from the rest of the viral caps and moved into our reaction tube. This ensures we have a pure sample of the virus's genetic code, ready for the conversion process. Now that we have isolated the viral RNA, the actual process of reverse transcription begins. We introduce a special enzyme called Reverse Transcriptase. Think of this enzyme as a molecular architect. Its job is to read the single-stranded RNA template and begin building a stable, complementary DNA strand, or cDNA. This is the RTO part of our RT-PCR process in action. As the reverse transcriptase enzyme travels along the strand, it synthesizes a complementary DNA sequence, or cDNA. Watch as it adds individual nucleotides to create a temporary RNA-DNA hybrid molecule. This step is absolutely essential, because standard PCR machines are designed to amplify DNA, not RNA. We have now successfully converted the viral message into a stable format. In the final step of reverse transcription, the original viral RNA is degraded and removed. We are left with a single-stranded cDNA molecule. This DNA template is now stable and ready for PCR amplification, where it will be copied millions of times for detection. Watch our previous video on PCR to see exactly what happens next.
Thank you for watching. Please like and subscribe for more valuable science lessons by Hima Learning Academy, where learning science is made easy. You can find my full PCR resource pack on my Teachers Pay Teachers store. The link is in the description.